Hi, I'm Morgan Johannesen, and I'm a senior in chemistry who also recently graduated with a BS in geology. And I'm really excited to tell you about the research I did under the guidance of Dr. Alison Olcott. I was able to examine the preservation of life in gypsum from Great Salt Plains, Oklahoma, which can be considered a modern analog or comparable environment to what Mars would have looked like a couple billion years ago. So why look for signs of life in gypsum? The first and most exciting answer is that it can help inform our search for life on other planets. Mars has an abundance of gypsum, a hydrated calcium sulfate mineral that forms when water evaporation drives up the saturation state of calcium and sulfate ions enough to cause it to crystallize out from the water. If we can detect chemical traces of life in gypsum, such as by identifying hydrocarbon biomarkers that may be preserved in it, or otherwise probe it for properties that indicate the presence of microbial life and organic matter in the environment at the time of crystallization, that can give us a framework for what to look for on Mars. Mars used to have a lot more water, and at one time had evaporative closed basin environments much like those on Earth. Additionally, since the gypsum at Great Salt Plains is currently growing, investigating how and if life might be preserved in these rocks can give us a modern baseline for comparison with much older gypsum in the geologic record. Great Salt Plains, Oklahoma is a unique extreme environment as a terrestrial inland salt flat that receives a great deal of solar radiation during the day and has high evaporation rates that peak in summertime. It was funded as an NSF microbial observatory that focused on documenting the diversity of the halophilic or salt-loving microorganisms that survive and thrive at locations shown with blue and green markers on the map. The groundwater gets its extremely high salinity by traveling through and dissolving subsurface Permian evaporite salt layers. Biomarkers were recently identified in some of these salt layers, specifically the Permian Blame Formation and the Flower Pot Salt to the north and west of this area, in Drew Lockamy's graduate work in Dr. Olcott's lab. The groundwater flows upward from these Permian salt layers through the overlying shale due to hydrostatic head and continues its upward journey to evaporate out at the surface of the salt flats via capillary action. I was able to go collect gypsum crystals, groundwater samples, and halite crust samples from the public crystal digging area at the Great Salt Plains a couple years ago. This area is free and open to the public from April to October every year. The picture on the left shows where we found these larger gypsum crystals in twinned clusters just about 10 centimeters below the surface. We also picked up lots of little equine and lenticular gypsum crystals that were left behind by previous visitors on the surface. You can see them scattered and partially buried in the sand and halite to the right in the foreground. Robert D. Cody and A.M. Cody at Iowa State back in the 80s and 90s investigated the chemical influences on gypsum growth forms in lab experiments. A major conclusion from their research was that organic acids tend to interact with the calcium 2+ cations that are exposed on the gypsum crystal lattice as it's growing, especially on the E and L faces, which causes these faces to grow slower and expand in surface area instead. Gypsum A on the left has a typical habit, while gypsum B on the right was grown in the presence of organic acids. The Great Salt Plains gypsum crystals have a type B habit, indicating the presence of organic acids in the sediments where they grow. This type of gypsum morphology has also been observed in Gale Crater on the surface of Mars. However, chemical analyses of these spots showed that these are actually aluminosilicate clay pseudomorphs, or replacement fill-ins of what was once this lenticular type B gypsum. Linda Kay and coworkers interpreted these as the diagenetic remnants of lenticular gypsum that had grown in the shallow subsurface due to the evaporation and capillary removal of water through the overlying sediments, likely in the presence of organic inhibitors, much like the gypsum growth conditions at Great Salt Plains. Only a few studies using fluorescence microscopy to examine microbial preservation in young and old gypsum have been published to date. A couple leaders in this area are J. William Schoaf and colleagues at UCLA and Kathleen Benison at University of West Virginia. In Dr. Olcott's lab, we have an Olympus BX51 fluorescence microscope that has both reflected fluorescence along with traditional optical transmitted light capabilities. We have an ultraviolet or UV excitation filter and another in the blue-violet range. I examined both the outer surfaces of the crystals and the insides by carefully slicing about a 1 millimeter thick shard from the B face, as shown under UV to the right. The other portion of this project involved analyzing biomarkers preserved in the gypsum, which required me to extract the organic components from the rocks. To do this, I weighed out about 100 grams of gypsum crystals, crushed them up with a ball mill, freeze-dried them to remove water, and then extracted the neutral lipids in the organic solvents dichloromethane and methanol with a microwave system that heats the samples in a controlled manner. Next, I vacuum filtered the lipids from the gypsum and sediment and evaporated the solvent off. Then I redissolved the dried lipid extracts in hexane and finally analyzed them with gas chromatography mass spectrometry, or GCMS. The microorganism I most frequently identified in the gypsum were these pennate diatoms in various degrees of preservation. Shown at the top are some other microscope images of diatoms that were collected at the Great Salt Plains to show what these unicellular microalgae with silica cell walls look like when they're alive. Image A was found on the outside of a gypsum crystal, and image B is actually from a groundwater sample in a petri dish stuck under the UV, and the rest were taken from B-face cleavage shards. Images A, B, C, D, and F were taken under UV, while E and G were taken with optical transmitted light, and image H was taken under blue-violet excitation. These are some carotenoid pigments and a carotenoid-producing filamentous microorganism to the right. On the left, images B, C, and D were found on the outside of the gypsum crystals, while the rest are from the interior shards. Carotenoids are naturally produced by microorganisms as light-harvesting pigments and as protection against photooxidation. These compounds fluoresce in the visible range, so they show up as bright orange-reddish under UV and the blue-violet excitation here. The second most frequently encountered microorganism in the gypsum next to the diatoms were these mineral-encrusted filamentous cyanobacteria. These are the preserved remains of Geitleranema, Lyngbia, and other cyanobacterial taxa that grow in the Great Salt Plains soils, some examples of which are shown at the top. Images A through I are all from B-face cleavage shards, and image J has arrows pointing out some cyanobacterial filaments on a whole crystal near the edge of an L face. I also observed a wide variety of fluid inclusions in the gypsum crystals, but some of the most interesting ones were along the basal cleavage plane with these tiny specks fluorescing various colors, shown here under blue-violet excitation with the white balance adjusted in the microscope software for better color resolution. These are most likely halophilic prokaryotes that could be taking advantage of the pockets of water trapped in the gypsum as it crystallizes. This could be an adaptive strategy in hypersaline evaporative environments when everything is drying out around your cells. Finally, here are some preliminary gas chromatography mass spectrometry results from the gypsum samples. Shown here are extracted ion chromatograms of isoprenoids and N-alkane fragments from 3 sets of gypsum samples from 10 centimeters below the surface. This gypsum had inclusions of a dark, organic, rich-smelling mud layer, and some had half mud, half sand inclusions as shown to the top left, and all had these various type B habits. The M to Z, or mass to charge ratio, of 85 is associated with fragments of alkanes, such as the C17 alkane shown to the top right. The M to Z 113 fragment is characteristic of isoprenoids, which are made up of many 5-carbon isoprene units. An example C20 isoprenoid known as phytane is shown to the right of the MZ113 chromatogram. Currently, without selected ion scans of specific mass fragments like these and comparison to standards, all we can say from this is that it confirms the hydrocarbons are being preserved in the gypsum, and the lipid profiles were quite similar across the different gypsum samples. The main conclusions I was able to make were that the gypsum growing in the shallow subsurface of the Great Salt Plains indeed captures a variety of microorganisms and biological materials as I observed with fluorescence microscopy. Although unfortunately cut short, the bit of GCMS data I managed to obtain confirms that organic hydrocarbons are being preserved in the gypsum. This has implications for target areas for searching for signs of life on Mars. The Jezero crater that NASA's Perseverance rover recently landed at also has remnant evaporative closed basin environments like those observed on Gale Crater. And if this gypsum were to be found on Jezero crater, I think it would be worth investigating. There is a lot that can still be investigated regarding life preservation in the Great Salt Plains gypsum and in gypsum in general. The biomarker GCMS results I got at the beginning of March 2020 were very preliminary and did not lend much insight for other lipid classes such as fatty acids and steroids that may be preserved in the gypsum. This is only because the most volatile lipids that I extracted were readily analyzable with gas chromatography, without forming more volatile derivatives beforehand, such as fatty acid methyl esters. The groundwater samples also, that I collected during fieldwork, have been held in a freezer at -20 degrees Celsius in glass bottles for future analysis. And as you can see to the right, the groundwater has an oily sheen to it. So the water chemistry as well as possible biomarkers in the water could be analyzed, which would make for an interesting comparison with the biomarkers in the gypsum. This research was partially funded by the KU Center for Undergraduate Research through an undergraduate research award that I applied for in the spring of 2018. And the fieldwork portion of this project was made possible by the KU General Research Fund awarded to Dr. Alison Olcott. I would also like to thank Dr. Alison Olcott for being a fantastic mentor throughout my entire undergraduate experience in geology and chemistry, and a great research mentor. And I'd also like to thank my friends Maya and Ian for helping me with fieldwork. And I'd like to thank you for watching my presentation. Thank you.